Welcome, ladies and gentlemen, to this lecture, a continuation of muscle physiology. We've covered most of the histology. We are looking now at skeletal muscle contraction. And pertinent to this, crucial to this discussion, is for you to understand the structure of the muscle fiber. Basically, the cell membrane, first of all, has got portions that invaginate or develop inside to wrap around the myofibrils. As it does that, it comes in contact with the sarcoplasmic reticulum on either side. This creates what is called a triad. A triad, tri meaning three. So three means we have the T-tubule, then we have on either side two cisterns in terms of storage units, cisterns like your water cisterns. So what do they store? They store calcium for the skeletal muscle fiber. Therefore, when the depolarization takes place on the surface sarcoplasmic reticulum, it spreads quickly into the muscle fiber and directly activates the release of calcium from sarcoplasmic reticulum. The calcium that is important to coordinate, to connect the excitation and link it to muscle contraction. Therefore, we say calcium is important for the excitation contraction coupling, meaning it is the main link between the muscle being excited by the motor neuron and the muscle being able to contract. Without this, there is no link, there is no muscle contraction. That is why calcium levels in the body are supposed to be in a particular concentration level for the physiology of the muscle to be sufficiently adequate. So that done, we move to the issue of the myofibrils. So the myofibrils that are wrapped around the sarcoplasmic reticulum and the T-tubules and the triads, these they are actually bundles of myofilaments. The myofilaments that are thick and thin. The thick filaments are actually the myosin, and the thin filaments are the actin. So specifically for myosin, it is made up of 6 components. So it is said to be an hexameric protein. So it has 2 heavy chains and 4 light chains, meaning 2 pairs of light chains. The first two are control light chains, which are regulatory light chains, and the next two are the essential light chains for the contraction properties of the muscle. So then we go to the actin. The actin is structured in such a way that, yes, it is called a thin filament because it is lighter. The myosin is made up of a 6 component structure, but this one, pay attention, it has the G actin or globular units, the spherical protein connections we are seeing. Then it has this rope-like structure that wraps around these proteins, which is called the tropomyosin. Then it has the troponin complex. The troponin complex together with troponin are 2 control molecules of the actin. These 2 molecules are important for skeletal muscle contraction. If they are not active, no contraction. If they are active, then there is contraction. Then what happens? What happens is that calcium, when it is released, it will bind to troponin. The troponin has troponin I, troponin C, and troponin T. The troponin C binds calcium. Then it becomes active and activates tropomyosin. Okay, the troponin C. The troponin C activates the troponin T rather. Troponin T activates tropomyosin. Then the tropomyosin flips changes shape and flips and exposes the binding site for myosin heads. And then the myosin heads will bind there and begin to pull the actin towards the center of the sarcomere. So in terms of summary, what happens is that, like I've said, calcium will be released, and it as a result of the nerve impulse, and this leads to troponin being activated. Then the troponin activates tropomyosin, then the tropomyosin changes shape and exposes the binding sites of myosin on the G actin component of the thin filaments. So basically, using the sliding filament model, this is what we are explaining here, that first of all, the ATP should bind to the myosin heads, such that the myosin heads will convert this ATP into ADP, and it will become now active to bind to the actin. When this happens, the myosin heads bind to the actin, forming what are called cross bridges. When this happens now is that the inorganic phosphate will be released, and the ADP together with myosin heads will become more active, and that will cause a stroke for the myosin heads, and you see that pulling effect. When this happens, the next stage is now another molecule of ATP to bind to the myosin heads and release the myosin heads from actin, and that is now relaxation. Therefore, what you learn from here is that ATP is needed both for muscle contraction and muscle relaxation, such that if someone dies and they are unable to produce obviously energy, then muscle relaxation cannot take place. Therefore, the muscle remain in a stiff state, and this will be called rigor mortis. And so we are saying in summary that muscle relaxation should be as a result of the fact that the calcium that activated troponin should be pumped back into the sarcoplasmic reticulum, and this will reduce activity of the control proteins, which is troponin and tropomyosin, and this will reverse the contraction and will go into relaxation. Best wishes in your exam. May God bless you. Bye bye.